Okay, we are live on YouTube. Fantastic. All right, so I think we'll just wait about 3 minutes, give people time to join in. Adam, have you been?
Yeah, all good. So same as
Same old.
all the weeks merging into one at this point. How's everything your end?
Good. Lots to do. I'm leaving
Signed your contract now,
tomorrow.
so
What's that?
you're leaving tomorrow.
Oh, I'm leaving tomorrow for a trip. Yes, my first outing into the world since the pandemic began.
It's just outside your
driving.
house.
Yeah, basically. I'm driving with my parents to Missouri, which is, I don't know if you know much about the geography of the United States, but it's a long way.
Somewhere in the middle, right?
It's basically, yeah, it's like the geometric center. So.
Why are you going to Missouri?
Fun. Oh, my older sister is graduating with a master's degree in social work that she's been getting on the side. So it's been a journey. She's worked super hard. I'm really proud of her. Yeah, so it's an in-person graduation. It's like, it's this weird thing where they have everybody. Yeah, but they'll have people be in pods, like in separate physical groups, depending on, who you're there for. I don't know. It might be strange. So we'll be driving through a lot of the country that maybe has different feelings about things like masks than I do. So it might be stressful.
Yeah. That's good that's in person. Virtual. I've seen virtual graduations last year and they looked awful.
Yeah, I mean,
I saw somewhere you have to make like an emoji of yourself and you have to like walk up the stage and like, shake hands.
Yeah, I saw a video of some guy like he had his virtual graduation up, and then when they called his name, he just walked by his TV, to recapitulate that. But yeah, it doesn't quite work.
I'll leave it to you to get started whenever you want.
Okay.
Mourino's in the chat somewhere.
Yeah. Okay. Marino, do we want to make sure your sound and are you there? Okay. Just making sure you weren't a ghost. Yes. Yeah. Hi, how are you today?
Good.
Okay, great. Thanks to you both for being here.
Thank you.
We are glad to have you. All right, so since we have two speakers, in the interest of time, I'll go ahead and get started, even though it's in my nature to always delay things. So our first speaker today is Katy Rothenberg. She got her undergrad and grad degrees in biomedical engineering from Boston University and Duke University. She's currently a postdoc in the lab of Rodrigo Fernandez Gonzalez at the University of Toronto, where she's studying adhesions in the cytoskeleton during collective cell migration. So we're really excited to hear about her current work today. And Katy, the floor is yours. Thanks a lot.
Okay, great. And let me just go back to my title slide. Okay. Yeah, good morning, everyone. Thanks so much for this opportunity to tell you about my work in the Fernandez-Gonzalez lab, looking at the small d2-PAs RAP1 during collective migration. And yeah, so in our lab, we're interested in these collective migratory processes, and in particular, the molecular mechanisms that help drive forward motion of cells. And so one of the model systems we use is wound closure in the Drosophila embryonic epidermis. And we like using the Drosophila embryo because it's very amenable to genetic, pharmacological, and biophysical techniques. And it's also optically clear, which allows us to do kind of some high resolution imaging. And so this is just a section of the embryonic epidermis about midway through development. You can see that the cells are outlined here in magenta, and that's a label for E-cadherin, a component of the adherence junctions. And then in green, we have the non-muscle myosin 2. And so the way we study collective migration in the system is that we use a high power laser to ablate 3 to 4 cells in the epidermis. And then we watch kind of as the cells organize the leading edge and then move forward to close this gap. And this is happening without any cell division, so it's purely a migratory process. And so as you can see here, as the wound is closing, the cells are moving forward. We have polarization of myosin to the leading edge, and that creates this supracellular actomyosin cable that's contracting, which helps to kind of coordinate the leading edge as well as draw the wound closed. And what you might also be able to see here is that E-cadherin is depleted from the leading edge and accumulates at these puncta around the wound. And those are the sites of the former tricellular junctions. So just kind of to summarize this process in graphical form, we have these adherence junctions, and this is what we know so far, is that the adherence junctions are endocytosed from leading edge, and then they reaccumulate at these former tricellular junctions. And then simultaneously, the cytoskeletal elements are polarized to the leading edge. And so we start getting assembly of this actomyosin cable, which then, starts contracting and starts driving wound closure. And we know that they're happening at the same time and they're dependent on each other, these rearrangements. So without removal of E-cadherin from the leading edge, we don't see the creation of this actomyosin cable and defects in wound closure. And similarly, contraction of the cable helps to reinforce these former tricellular junctions. So, these rearrangements are very interdependent. And then another interesting property about the cytoskeleton that we found is that heterogeneity at the leading edge is actually important, especially in the early stages of wound closure. So what we see is that we get kind of patches of actomyosin-rich areas. So here, this is showing F-actin. So we get patches of kind of high accumulation, and these patches contract and they strain the neighboring kind of actomyosin poor areas, which then induces further accumulation of cytoskeletal elements. So it's kind of like a ratcheting mechanism to ramp up cytoskeletal accumulation at the leading edge. And in mutants that don't show this heterogeneity at the leading edge and have a more homogeneous accumulation, we see defects in wound closure. So we do think that this kind of uneven distribution of the cytoskeletal elements is important for driving wound closure. And so what we know so far is that these adhesion and cytoskeletal rearrangements are important for driving wound closure, but we don't know what's upstream in kind of coordinating these rearrangements. And so I became interested in the small GTPase RAP1 because it's known to be responsive to some of the signals that we find at the leading edge. We have mechanical stresses, and we have presence of reactive oxygen species, which previous work in our lab has shown is important for allowing these rearrangements to occur and wound closure to happen normally. And so both of these are known to act on RAP1 activity, and then RAP1 in turn, in a variety of systems, has been shown to interact with cell-cell adhesions, cell matrix adhesions, and the cytoskeleton. And so I thought it was kind of a good candidate for something that may be integrating signals at the wound edge and then dictating these molecular rearrangements to drive wound closure. And so what I hope to tell you about today is what we found so far about how RAP1 interacts with cell-cell adhesions and the cytoskeleton to drive wound closure. So just an introduction to some of the genetic tools I'm using. I have a RAP1 dominant negative construct, which disrupts RAP1 activity. And then I have a constitutively active construct that increases RAP1 activity. And I use this UAS GAL4 system to spatially and temporally control expression of these constructs, specifically in these mid to late stage embryos. And so in our control embryos, we see this wound closure process happen. It's pretty stereotypical and happens pretty quickly. Within 45 minutes to an hour, wounds are generally completely closed. And again, you see this nice cytoskeletal polarization. In the dominant negative, however, we see this defect in wound closure. The wounds are closing more slowly, visibly. There's also some defects that we can see in the tissue, which I'll get to more later on. But just looking at the wound closure rates themselves, we quantify this by looking at first the area of the wound over time. So on the onset of wounding, when we make the cut, since there's tension in the tissue, we see an initial recoil, and so an expansion of the wound. And then as we get these rearrangements happening, we see the wounds close in a fast phase first and then a slower phase. And you can tell from just looking at the area over time for the dominant negatives is that there's, first of all, a small delay in the onset of closure, and then it occurs more slowly. And we can quantify the rates of wound closure by fitting these curves to an exponential decay function and extracting the time constant. And when we do that, we can see that there is a significant difference in the closure rate constant in the dominant negative. So we're pretty confident saying there's a defect in the ability of the cells to move forward and close the wound. When we look at expression of the constitutively active, what's interesting is that we actually see acceleration of this process. So looking at the area over time, it looks, kind of a subtle difference, but the wounds are fully closed by about 35 to 40 minutes. And then when we quantify the closure rate constants, we do see a significantly faster rate of wound closure. And this is pretty interesting. We don't often see things that accelerate wound closure since it's already such a very efficient process. But yeah, so this kind of shows that RAP1 is being regulated during this process. It seems that its activity is important. And so the first thing we looked at to probe further into how RAP1 might be contributing to wound closure is first looking at the localization. And so, as I mentioned before, E-cadherin is depleted from the edge and along with other adherence junction components, and then accumulates at these former tricellular junctions in these puncta around the wound. And we hadn't quantified this previously, but now we've quantified it, and we see there's about a 20% increase in intensity at those, specifically at those tricellular junctions. And when we look at the accumulation of RAP1 in the same embryos, we see a very similar accumulation at those sites. Again, about a 20% increase. And so it looks like, RAP1 is going to these tricellular junctions along with E-cadherin. And to look at the timing of this process, we did a cross-correlation analysis between the two curves, the E-cadherin and the RAP1 curves, and found that the maximum correlation occurred with a time shift of 0. So meaning within our resolution of time, which is about 15 seconds, it looks like E-cadherin and RAP1 are arriving at these tricellular junctions at the same time. And so we started to think about what could be mediating this interaction. And we thought about canoe or ephadin in mammals. And so canoe and ephadin are known RAP1 effectors that also interact with adherence junctions. And so that could provide a potential physical linkage between the two. So when we look at canoe localization, we also see accumulation at the former tricellular junctions. So you can see these puncta that correspond with the E-cadherin puncta, a similar increase in intensity of about 20%, and they seem to be arriving at the same time. So, this is correlation showing that, all three of these components seem to be moving to these former tricellular junctions. And so the next logical step is to disrupt RAP1 activity and see what the effects on these rearrangements might be. And so the first thing you'll notice is that in the dominant negative, we do see kind of decreased E-cadherin localization to the cell boundaries in general. And that's expected. There's a lot of literature showing that RAP1 is important for formation and maintenance of adherence junctions. So that's not completely unexpected. But if we compare, relative to this initial intensity, and we look at intensity first at the leading edge of the wound, so at these linear junctions between the tricellular junctions, in both cases, we do see this loss of E-cadherin at the wound edge, which is what we expect from this endocytosis. But at the tricellular junctions, whereas in the controls, we get accumulation and we get those nice puncta, in the dominant negative, we actually see a loss. So there's no kind of reinforcement of these tricellular junctions. In fact, there's very little E-cadherin at the leading edge at all. And so this encourages us in thinking that RAP1 is important for localizing E-cadherin to these tricellular junctions. If we look at the constitutively active case, we don't see a big difference in localization of E-cadherin either at the linear junctions or the tricellular junctions. And so that process seems to be occurring normally despite overactivity of RAP1. And so that made us think that potentially there could be more to the story than just the rearrangement of the cell adhesions. And so we next moved on to look at the effects of RAP1 on the actomyosin cytoskeleton. And so here I'm showing some wound closure movies with a marker for F-actin. And in the control, what you can see is this nice polarization, accumulation of F-actin at the wound edge, and even some protrusive activity, especially towards the later stages of wound closure. In the dominant negative, we actually get this defect in actin accumulation. And we can quantify that by looking at the average intensity around the wound edge. And indeed, there is a defect in how much F-actin is getting to the wound edge. And so that could certainly affect the ability to create, a nice robust actomyosin cable. And then what's also pretty interesting is looking at the heterogeneity around the wound edge. As I mentioned, the heterogeneity is actually pretty important. We see a pretty big difference between the two. So in controls, so these are kind of chymographs of the wound edge, where the wound edge is linearized and then normalized to a specific distance. So we keep the same distance over time. And this is the first 15 minutes of wound closure. Normally what we see is that upon wounding, we get some accumulation of F-actin around the wound in those patches. And that's, in this plot, that spike in heterogeneity. And then over time, as those patches contract, they induce accumulation in neighboring areas so that eventually you get this kind of solid cable encircling the wound. And that's that decrease in heterogeneity. In the dominant negative, we do get those initial patches and you see a little bit of a increase in heterogeneity at the start, but then it remains static. There's no kind of induction of further accumulation in the neighboring regions. And so there's no reinforcement of the actin around the wound edge. So yeah, that was pretty interesting. And so, part of making contractile cable is also having myosin. So we also looked at myosin accumulation and saw a very similar effect. We see a defect in the accumulation, so we don't get as much myosin around the wound edge. It's very clear kind of in this 20-minute mark. And then again, we do get some patches of accumulation, but no reinforcement. And so we do think that RAP1 is important for reinforcing the actomyosin cable at the wound edge. Conversely, when we look at the constitutively active case, while we don't see, we don't see a difference in accumulation from start to end of wound closure, if we look at earlier time points, we do see that myosin accumulates earlier in the constitutively active than in the control. And so, even at 5 minutes, we're starting to get more accumulation here, which could explain how the wounds are closing more rapidly. And so kind of just a summary of what I've talked about today is that we think that RAP1 may be responsible for kind of the reinforcement of cell adhesion and actomyosin at the leading edge. Without RAP1 activity, we do still see kind of these initial rearrangements. We see the removal of E-cadherin. We see some polarization of the cytoskeleton, but we don't get this reinforcement of the tricellular junctions or kind of the responses to the forced transmission that's required to further polarize actomyosin to the leading edge. And so we think, based on our work so far and evidence in the literature, and we're working on confirming this, we think that RAP1 is contributing to the rearrangements of adherence junctions through its effector, canu or affidin. And we do think that it is having an effect on the actomyosin cytoskeleton. But since there's a lot of interplay between the cytoskeleton and the cell adhesions, we're unsure whether RAP1 is directly regulating the cytoskeleton or if this is occurring indirectly through, assembly of these tricellular junctions, allowing for assembly of the cable and, forced transmission. So these are things that we're continuing to look into and hope to have answers for soon. And so with that, I would like to thank my advisor, Rodrigo, and the rest of my lab for their support, as well as our collaborators for helping us out with some reagents and the resources we use and our funding, of course. And I'd be happy to take any questions.
Fantastic. We've got a lot of questions for you in the chat, so we'll try to go through them and get to as many as we can. So first, we actually have two from Mark Pfeiffer. First, does the RAP1 dominant negative, do those embryos eventually close the wound?
Yes, as far as I can tell, the embryos do eventually close the wound. So they may be relying on, whatever cytoskeleton they can get there, or they may be using, different techniques. For example, they may be using protrusive activity or something else to help draw the wound close, but they do eventually close.
Great. And the second one from Mark, have you looked in the same RAP1 dominant negative, looked at the effect on the CNO localization?
No, we have not looked yet at canu localization with the dominant negative. That's part of the experiments we have lined up to confirm that canu is actually the intermediate that we think it is. Yeah, that's a great suggestion though.
A couple of questions from Ankita. Is the myosin only planar polarized or also apically localized? And does RAP1 activate, feed into Rho to activate myosin?
Okay, so the first question was, is myosin not only planar polarized, but also apically polarized? Yes, so the actomyosin cable is at the apical surface of the cells. We don't know much about what's happening on the basal side in general, but yes, as far as we know, the cable is at the apical side at the same plane as the cell adhesions. And then the second question was, could Rho be involved? Right, that was the question.
Yeah, can RAP activate Rho, I guess.
Yeah, so there's some evidence in literature that RAP1 interacts with different Rho GTPases. Nothing's been shown in this system, but we do suspect that there is some sort of crosstalk between RAP1 and Rho, right, to promote this polarization. We're working on that now using a Rho activity sensor to see what's happening when we affect RAP1 activity.
Fantastic. All right, two from Ashrit. First, is the wound healing dominated by cell deformation rather than migration here?
The cells definitely are deforming. They're definitely changing in area. but they're also, moving forward into the wound through both displacement of their kind of forward and backward edges, and then there's also protrusive activity that enables kind of cell crawling. So yeah, I think it's a combination of the two. The cells are definitely stretching, but they're also actively moving forward.
Fantastic. And then on a related note, he says it looks like the cells elongate a lot at the wound site. Do they retain their regular sort of polygonal shapes after the complete wound closure?
Yes, so the cells do elongate since there's no cell division, the wounds have to be closed and the tissue returns to normal, lacking those original cells. So there is elongation, but they do eventually return to their normal shape. There's no scarring following the wound closure. So we haven't studied much how the wound resolves, but if you look far enough after closure, you can't tell that the wound was there.
Okay, and for the sake of time, we'll make this the last question, and then maybe we can come back at the end to answer the rest. From Amrita, who asks whether the canoe mutants are as severe or slow at wound healing as the RAP1 dominant negatives.
Yeah, that's a great question. So that's kind of next on my list of experiments. I'm kind of curious to see whether disrupting canoe, does it only disrupt the rearrangement of the cell-cell adhesions, or does it also disrupt, the cytoskeletal rearrangements? And I think that will help us tease apart how much feedback there is between the two and how independently RAP1 is regulating the adhesions versus the cytoskeleton. So yeah, it's a great point, and it's on the list.
Great. Thank you very much, Katie, for a very nice talk. Maybe we can come back at the end for the rest of the questions.
Yeah, thank you so much.
Okay, so we've got a second speaker now, who is Marino Arroyo, who is a professor at the Universitat Politecnica de Catalunya in Barcelona. He obtained his PhD in mechanical engineering at Northwestern University and a postdoc at Caltech. His research is focused on mathematical and computational modeling in mechanics and materials, and for the last 10 years has progressively turned into mechanobiology. Today he's going to be talking about the crawling of Euglena cells by large amplitude shape changes. So I'll leave it for you. That's Marino.
Okay, so thank you very much, and I'm really delighted to be here giving this seminar. So I'm going to start with my presentation, and before that, I mean before going to the minimize this to the main topic of my talk. In my group, we do a number we study a number of systems having to do with cell migration during development, homeostasis, and disease with wonderful collaborators. And all these examples and most of those that we have heard of in this fantastic seminar series that you have put together have to do with animal cells. And of course, animal cells are very important, but they are a corner in the tree of life, and there's a very big diversity in the life forms. You can see here that there's a diversity in the shapes of cells, and there's also a big diversity in the strategies that these cells use, for instance, to move around. And today I'm going to talk about how one mode of locomotion of Euglena cells, and I hope that this will be of interest to the audience of this seminar series, and I hope that the organizers will not consider changing the name to animal cell migration in this seminar series. Okay, so what are Euglena cells? So Euglena cells are a diversified family of unicellular protists that live in a variety of aquatic environments. And most of the times, so they are really common, and most of the times you will see them swimming using the flagella. These are examples by actually my collaborators, where you can see how they swim, they turn, beating their flagella. This is a very interesting system in its own regarding swimming. But they also do something else, at least a group of Euglena, which are this so-called euglenoid movement or metaboly, which are these highly concerted and very harmonious shape changes that that you can see in this movie that first fascinated us and prompted us to study this system. So in fact, if you want to look at Euglena in action, it's very easy because you go to YouTube, and it is a favorite system amongst microscopy aficionados, because as I said, you can find them very easily in ponds, all over the globe. And in fact, these kind of motions already puzzled the first microscopist that, arguably the first microscopist that described them as motile and green animalcules. And something that struck him was that they were so motile and also green. Now, there are other cells that move or crawl using large amplitude motions, but what's striking about Euglena is the fact that they do so in this very highly concerted and harmonious way. Okay, so what I'm going to talk about is biophysical approaches and combining theory and observations to understand how they do these motions and why they do it. Now, regarding how, this is a review, already a few years ago, and as you can see, as regards to metabolism, the cell structure, molecular motor, and motor category appear as unknown. So maybe this is a little bit of an overstatement, but actually the molecular motors are still unknown. But the cellular structure needed is well understood, and it's the pellicle, which is this striated cell envelope that these cells have. So these are a variety of euglenids, and a few of them have large numbers of strips, and these ones usually correlate with those that have the ability to change shapes. So these one are, for instance, rigid. So how does this pellicle work? So this pellicle is a very interesting organelle that is below the plasma membrane, and that is composed of proteinaceous elastic strips adjacent to each other that are bridged by microtubules and tubes of endoplasmic reticulum that are involved in the regulation of their motion. And right here at these junctions between the strips and next to these microtubules, there are molecular motors that haven't been identified that enable the relative sliding of these strips, which results in a shearing deformation that integrated at the cellular level results in shape changes. Okay, so let me explain this a little more. So one way to think about it is imagine that you have an idealized cylindrical cell with these strips that can slide relative to each other. So just for the sake of the thought experiment, imagine that you open the surface and then you slide all these strips uniformly, and then you rotate it and roll it back again. So you see that by sliding, by shearing these strips, you accomplish a shape change, in this case with a shorter and thicker cylinder that has the same surface area. Now, strip sliding induces local strain and shape changes. Now, what happens if strip sliding is not uniform? So I could show you what happens with equations, but I can also show it with a system, with a model, with a plastic model, in which you have strips, and you can see that can slide. That is a 3D printed model, and you can see that when this sliding is non-uniform, you can accomplish very large amplitude shape changes, and you can see also how cellular deformation is correlated with rotation. And we not only did this model to explain the mechanism of cell morphing in Euglena, but also because we think that this is an interesting engineering principle to morph surfaces. So this is an example where local shear results in a local strain, and this local strain is heterogeneous and results in and necessarily requires this shape change. And there are other instances in which there is morphing of a thin sheet by non-compatible strain. So one example is growth. So growth, you can understand as strain. So if growth is uniform, then an object just becomes bigger. But if it's non-uniform, and non-compatible with the original shape, then it will lead to morphogenesis. And this idea is also used in man-made systems, traditional ones where the crocheting knots induce a non-compatible strain, or in technologies for shape morphing. Now, let me give you 2 slides before I go to really observations about these cells undergoing these motions, about the mathematical modeling of this pellicle. And then, so there's a very elementary theory that allows you to understand these 2 problems. So first, how giving a non-uniform sliding distribution, so how these adjacent strips slide across the pellicle surface, you can find the resulting shape, but also this inverse problem by which, given a shape, looking at the shape, you can infer what was the required sliding distribution, okay? And thinking about this mathematical model, you can understand that this is really a flexible, a powerful mode of shape morphing. So you can take a cylinder and transform it into a sphere, into a disc, into a cone, into many other shapes. And these shapes look very abstract, but you can see that if you stitch them, you start getting what these cells are doing. Now, you can also use this theory as some kind of mathematical microscope that will allow you, from the observations of the shape changes, to infer what are these shears in space and time, and then to infer, for instance, what is the amount that these adjacent strips are sliding and what are the velocities. So even though we don't know what motors are causing these shape changes, we know that they can move at about half a micron per second, which is compatible with motors, processive motors running along microtubules. Okay, so in a way, this pellicle is like an extended axoneme. So the axoneme that you find in cilia and flagella is this structure of microtubules and molecular motors. So imagine that you extend this to a surface, okay? So in the same way that the sliding between these tubes, between these microtubules, leads to the bending and the twisting of the flagellum, then the sliding of these motors leads to the shape changes in the Euglena pellicle. Okay, so I told you a little bit about how are these shape changes accomplished. So now let me tell you a little bit about, well, I told you about how, but again, these motors are not known, and there's very little known also about the regulation of how these motors are regulated. So now let me tell you a little bit about why, or how we try to understand why they do it. So in the biophysical literature, one perception was that these motions, these large shape changes, were a means of swimming into a fluid. And in fact, Euglena inspired a prototypical mathematical model for swimming. So we also examined this, and indeed we found that this is a competent mode. So this motion is non-reciprocal, which is what you need. So it's not time reversible, which is what you need to be able to move without inertia. And in fact, the light hill efficiency is reasonable. However, this is not very convincing because the speeds are about 100 times slower than flagella swimming. Okay, so these cells also have flagella; they swim very efficient with that. So why would they need this complicated, motile organelle if they already swim very well with the flagellum? Now, some of these euglenids are phagotrophs, so they can engulf large prey. And in this case, it is accepted that they need the plasticity of this cell surface to be able to engulf large prey. And here I'm showing an example of a Euglena cell feeding on another eukaryotic cell. Yet, in many of these other Euglena cells that perform metaboly are osmotrophic or autotrophic, so they don't engulf large cells. And in this case, it is viewed that the ability to perform these large amplitude motions is an ancestral vestige, maybe without a particular purpose. Yet there is circumstantial evidence in the literature that suggests that this metabolism may be useful to move in confined environments. And there's a number of such circumstantial evidence in the literature. However, the one I found more interesting was again found in YouTube, and again it's one of these microscopy aficionados that caught this Euglena moving in a dense bacterial suspension. And here you can see that the flagellum is really not useful, whereas this crawling motion seemed to allow this cell to really move in this difficult environment. You can also see that whenever there's a little bit of space, then the flagellum seems to work, but then again, when it gets caught, crawling will continue enabling the motion of this cell. So to study this more systematically, we looked at cultures of Euglena gracilis cells between glass plates, and then what you see is that when you have a dilute culture, you see this characteristic flagellar swimming. However, when you have a crowded Euglena culture, you see that there are like contacts between these cells, and you see a variety of behaviors, which include rounding of these cells and spinning. And then you see also examples of metabolism, cells undergoing these peristaltic motions, although this doesn't seem to really help them to move in this setting. Now, if you consider glass plates that are closer together, so that the cells become really confined, then cells systematically starting developing these large amplitude motions metabolism. And furthermore, this setting allowed us to watch, simultaneously the reconfigurations of these pellicle strips, so how they change in configuration and the motion of the cells. And in this case, as I'm saying, confinement led to the triggering of this behavior, which seemed to propel the cell forward during these motions. So this setting also allowed us to further understand quantify this sliding between the pellicle strips. So an elementary theory shows you that the curvature, because these strips that I'm seeing lie on a plane, you can correlate the curvature, you can identify the curvature of these strips with the rate of change of sliding. So this is a pellicle shear, the sliding between these adjacent strips as a function of arc length. And then by looking at a strip at different time instants, this allowed us to compute the sliding in microns, so to quantify these excursions of about 1 micron in relative motion of adjacent strips, and again this sliding velocity of about 1 micron per second. Okay, to simulate in control conditions a multidimensional confinement, we drove these cells into tapered capillaries, and this is what we found. We found that when there was sufficient space, they would swim. However, when the space became narrower, they started colliding and exhibiting these vacillations with initial shape changes. We also found that this rounding that we saw in the dense cultures was also present in these confined capillaries and seemed to allow cells to turn in this capillary. And more strikingly, we found that eventually nearly all cells develop metabolism. And here we were struck by how this stereotype behavior seemed to acquire a clear utility in locomotion. And this was enabling cell motion even when the degree of confinement was very significant. So looking at this, we looked a little more about the power phase. So when this bulge moved backwards, and we saw that it establishes contact with the capillary walls, and then as it propels backwards in the frame of the cell, this propels the cell forward. And the same thing happens when you have the same stereotype behavior can be observed when you have a higher degree of confinement. However, in this case, there's a larger part, region of the bulge that is in contact, and maybe is in contact for a longer time. And then, of course, as confinement proceeds, what happens is that the shape excursions become narrower. So you see that there is a trade-off between the degree, between being too loose and establishing contact seldomly with the boundaries, and being too tight and have the chance of have slack to have very little slack for shape excursions. But other than that, the mechanism was seemed to be the same across confinement. And again, in these chymographs, you see very well the overall forward motion of the cell and the backwards bulge motion during the power phase of this crawling mode. Now, we looked at how the period of this stereotype behavior depended, and the cell velocity dependent on the capillary diameter. And we found that whereas the period of the cell gate, which is about 8 seconds, was very insensitive, quite insensitive to capillary diameter, the normalized cell velocity depended very much, and and had a maximum for a normalized capillary diameter of about 1.3, which is this degree of slack of cells relative to the capillary. Okay, so these are normalized cell velocities. If you look at absolute cell velocities, these cells move at about 2.5 micron per second. And so is this fast? Well, a few years ago, there was this paper that was the first world cell race. And these are animal cells that are adherent animal cells. And you see that the faster cells were about 1 to 2 microns per minute, whereas this is per second. Now, later, when you are in a confined environment, animal cells can move faster, up to 10 microns per second. So, with this slide, you can see that if Euglena were allowed to participate in this race, it will rank first. Okay, so the conclusions so far are that confinement triggers metaboly, that metaboly is an effective and fast crawling mode of locomotion under confinement, and the confinement flagellum beating doesn't work. And having understood this, we then ask the question of, what are the forces? What is the mechanism for locomotion during metaboly? And for that, we took as a background what is known for crawling of animal cells in confined non-adhesive environments. In this case, it is well known that cells can become polarized. So the actin cell skeleton can become polarized. And then the retrograde flow can mobilize frictional forces that will propel the cells forward. And of course, if this is a capillary or a channel, then this motion will be opposed by a hydraulic resistance that, depending on the setting, can even lead to stall of this motion. So you have a propelling force, which is due to this friction of the cortical layer relative to the capillary, and then you have this hydraulic resistance that opposes motion. Now, this looks in many ways, so the Euglena looks in many ways similar to this setting, because during this power phase, by moving backwards this bulge, you are moving backwards cortical material that is becoming in contact and possibly mobilizing frictional forces. So we wonder whether this analogy, how far this analogy went. And for this, we developed a minimalistic mathematical model of the power gate, of the power phase of this gate. So this is again an idealized cell, a cylindrical cell, and now we have a pulse of a wave of shearing that causes this bulge and that moves backwards as we observed in our experiments. Now, if you assume first, for the sake of discussion, a limit of high friction, so like in a bicycle where you are rolling and the tire is not slipping with the road. Now the tire is this bulge. Then what we found is that indeed this was sufficient to propel the cell forward. But something that was very remarkable is that when this happens, the fluid is pumped backwards. Okay, so the cell is moving to the right, and instead of pushing fluid to the right, as an animal cell will do, the cell was pumping fluid backwards. Okay, and of course this is due to the fact that when the cell does this, it is also changing shape. Okay, so this peristaltic motion allows it not only to move in this direction, but also to pump water in the opposite direction. Okay, so this led us to hypothesize that that pumping of water could be a means of propulsion. So not only friction with the capillary, but also pumping of water could be a means of, through this peristaltic motion, could be a means of moving in this confined environment. So we consider the limit of high hydraulic resistance. So now there can be no fluid flow across the cell. So it's like a plug. And we found that these cells were, again, moving forward without any problem, in agreement with this hypothesis. And then you can consider, the intermediate regimes between the high friction and the high hydraulic resistance. And you see that in all cases, the cells will be able to move forward, unlike an animal cell that would become stalled in the high hydraulic resistance regime. Okay, so the theory predicts that these confined cells can move without inducing any flow rate in the capillary. Okay, so we turned to the experiments to see whether this was true, and indeed we found that a number of evidence that show that these cells were able to move and crawl without disturbing the fluid. This is an example where you see that that there is one cell that is stuck, and with independent experiments we show that these cells behaved as hydraulic plugs, and next to it you see that this cell is able to move forward, right? So the water here is incompressible. Okay, so this water is not expanding. It's just that by this hydraulic, by this peristaltic motion, the cell is also transporting water from here to here as it crawls. And the same thing was observed when looking at tracking fluorescent beads. Okay, so the conclusions so far are that what I said earlier, and also that that this mode of locomotion is robust and adaptable with respect to the degree of confinement. As we've seen, they're able to crawl in very thin capillaries, and also to the mechanical nature of confinement, whether it's hydraulic or friction. Now, the last thing that we asked, and that I'm going to talk about, is tries to address the following question, and it's whether this complex and adaptive behavior by which these cells are adapting the gate to the degree of confinement required a dedicated mechanosensing and mechanotransduction machinery, or instead whether the active pellicle was able to mechanically self-adapt to this changing environment. And for this, we developed a computational model that viewed this active surface as an extended elastic and motor clutch system, where locally we modeled a constitutive relation between the velocity of motors and the force that they feel following this linear relation. And then we assumed a given activation of these motors with a force-free velocity, that depended on space and time, that would produce a free, a cell deformation similar to those observed in a free fluid. And then we considered the frictional forces with a capillary, the hydraulic forces, contact forces, cellular pressure, and the fact that this membrane is locally shearing according to these active motors. So when we did that, we found that we basically were able to recapitulate all the observations, and we saw that this system, without having to explicitly model any mechanosensing and mechanotransduction, but rather by the mere biophysics of this envelope and the fact that the motors can slide when they find, they can stall and slide when they find too much resistance, we're able to find this gate adaptation depending on the degree of confinement. And when we look at these chymographs, or how the normalized velocity depends on capillary diameter, we remarkably recapitulated the observations that we had on the experiments. Okay, so I add one more point in my conclusions then, which is that the pellicle of euglenids is an, at least our theory suggests that it's an extended and elastic motor system that, once biologically activated, can mechanically self-adjust to the degree of confinement. And if you look at this from the soft robotics point of view, this is an instance of what is called mechanical or embodied intelligence, by which you outsource the difficult controls of a mechanical task to the compliance of the materials that make this soft robot. Okay, so to some degree, we think that Euglena represents an example of this mechanical intelligence. Right. So let me give you, as an outlook, a question that we asked ourselves and briefly reported in this paper. And so one question is, whether these cells really take advantage of this capability, in their native environment, which is an open question. Then another question that we have is how this, biophysical characterization of metabolism can help us, can help us, I don't know if understand, but, suggest questions within the evolutionary history of Euglena cells. And in particular, how and why these cells retain this active pellicle, which, as I said, is a vestige of phagotrophic ancestors, and develop the ability to operate this machine that, for a phagotroph is used for feeding, to operate it in a new way, in a non-reciprocal way that is compatible with crawling. And to try to shed some light into this question, we looked at other species in confinement and devoid of confinement. So we looked at a phagotrophic species, which is Peranema, which in a surface is known to glide, thanks to the flagellum, looking for prey. Right? It's a very, I would say, aggressive cell that will feed on means looking for some cell to engulf. Now, under confinement, metabolism is indeed triggered, but as you can see, it doesn't seem to be really non-reciprocal and useful for locomotion, right? So in this case, this suggests that the ability to deform the body in this cell is used for feeding, but maybe not so much for this crawling by metabolism. On the other hand, we looked at another species of non-phagotrophic, in this case an osmotroph, which is the Stigma proteus, and we saw that when it was not confined, it swam readily and quickly. However, under confinement, again, it was able to develop this peristaltic motions that led to locomotion. Okay, so in some sense, this suggests that these cells, these cells that have phagotrophic ancestors, developed, an innovation that was not, a new cellular structure, but just the regulation that allowed them to use it in a different way, in a non-reciprocal way that is compatible with crawling. And with that, I will end and thank my group and in particular my collaborators in this project that are Giovanni Noselli and Antonio De Simone, that have been collaborators in this project for a long time. And as a bonus, I wanted to show you this other instance of another non-animal cell that exhibits really funky and interesting, cellular shape changes, in this case for feeding, not for locomotion, maybe. And just to suggest how interesting it can be to look at cell locomotion between beyond animal cells. And with this, I will stop and welcome questions.
Oh, fantastic. Thank you so much. And you're absolutely right. And that we have definitely been sort of focused on just one small part of the tree of life. I'm wondering about the control decisions, I guess, right? So you've got these, they're swimming along, and it seems like they sort of, if they're in an open space, prefer to use the flagellum. And then, how are they detecting that they're confined? Is it curvature, or somehow this organism needs to sort of make a switch in its behavior, and how is that happening?
Yeah, so as far as I know, it's not there's not it's not completely clear, but all evidence suggests that it is contact and collisions, and that maybe it's the flagellum, the forces that the flagellum feels from obstacles that will trigger these are calcium waves in these tubes of endoplasmic reticulum that will trigger this action of these molecular motors in this extended motor system. But it's really not known. The molecular tools to play with these Euglena cells are very limited at this point.
Thanks.
Fantastic.
Love those movies. Those are really cool shape changes. Ricard Alerts asks if the strips can slide without bending, or does sliding always come with bending?
Yeah, so it always comes with bending because they are constrained, right? So they are, because of the geometry, they cannot freely slide without bending. Now, I guess that if you were to cut the cells at the poles and activate the motors, which in principle you can do, these cells can be reactivated, they can be killed. So you treat them with detergent. People have done it, and then you end up only with this machine. You activate it with ATP, and then you can contract it. So I guess that if you were able to cut these poles, then they will slide without. I mean, you could expect to see that they slide without bending.
So it's essentially because the strips go from end to end of the cell, and they never.
Yeah. Okay. Yeah. So they're constrained, and there's no way that they will be able to slide without bending.
Thank you.
Great, thanks a lot. So from Nath House says, very interesting talk, Mourino. Do you have any other animals developing similar motile coat? For example, dictyostelium. Have you tried any drugs targeting microtubule dynamics?
Okay, so I think that this particular mode of mechanism of shape changing is quite unique in Euglena. So there may be other cell motions that share some features, and in fact maybe these lacrimaria cells share some features, but they are distinct. And no, we haven't tried to apply drugs that will interfere with macrotubules or with motors with macrotubules. But what is known in the literature is that the motors that activate this pellicle are different from those that move the flagellum. So people have applied the drugs that impair flagellar motion, and still the pellicle was motile.
Great, thanks. Raphael Petrosian says, thanks for an interesting talk. Have you looked into how the cell will move if there's active flow of fluid in the opposite direction to cell movement? And how efficient is locomotion and metaboly in that case?
Mm-hmm. No, this is I mean, this will be actually interesting. The interaction between the flow and the crawling motions. What we did here is to use flow to move cells and to maybe take them to the tightest places of the capillary. And what we're able to check is that they are in these conditions they really behave like hydraulic plugs, so they are quite impermeable. However, how they will react to flows, we didn't look at, and it's an interesting question.
We have a question from Itaka. During the metaboly, intracellular organelles in the genome seem to be harshly squeezed. Is there any special mechanism to protect them from mechanical damage?
Not that I know. I mean, really, the inside of cells looks like a complicated mess. So not that I know of. Yes, it's true that there is an induced flow within cells and a steering of the cytoplasm, which has been also suggested as a benefit of metaboly. But as far as I know, there's not any other cytoskeleton that will give coherence to it, but rather all these organelles are being dragged by this active surface.
Do they have a nucleus?
Yeah, sure.
Yeah, okay. So, there's probably, do they have any of the same kinds of, lamins and things that are protecting the nucleus and, the cells that I'm used to looking at?
Yeah, I would guess so. I'm not 100% sure, actually. I would guess so that this is the case. Many of the organelles that you see are chloroplasts, actually. They are plastids.
Right.
Because many of these acquired, through secondary so basically they ate algae and endosymbiosis to develop this ability to feed on light. So many of these are plastids, and there are many other organelles that have been studied. But I am actually not an expert in the biology of Euglena, which is very interesting, and there's a community looking at it.
Perfect. Yeah, I think Mitaka's questions, because it's sort of come out recently that, the squeezing of, for example, cancer cells it turns out it's really important. So there's a lot of
Sure. It's an interesting question, yes.
Deepak, if you want to unmute yourself and ask your question.
Yeah, thanks for a very interesting talk. I had a question about the rotation of the cells that you mentioned, and whether that plays any role, or does it just disappear with confinement?
Yeah. Actually, the rotation. Here you see that some just round and spin, right? And then anecdotally, we find that in these intermediate degrees of confinement, they confinement.
Thanks.
Okay.
All right. Fantastic. I think that's okay. I had one more question. Sorry, coming from my complete ignorance about such organisms. I'm just wondering, are they we hear a lot about myosin and Rho and RAP and actin. Is it all the same subcellular components that are generating this motion, or is there is there unique machinery in these organisms?
Yeah, so I would say it's to some degree it's a unique machinery, because it's not actin. These are these are, let me see if I can make this bigger. Right, so these are microtubules here, right? And so one of these strips will end here and the other will start here. And, presumably there are motors, processive motors running along these microtubules, which have not been identified, okay? So this is definitely not related to actin contraction. And if anything, it is much closer to microtubule sliding in a flagellum. Okay, so it shares, resemblance and analogies with a eukaryotic flagellum, right? These are eukaryotic cells, but this, active cortical complex is original and unique, and this is what you find when you look at protists, that they are, so diverse, and each one find, one different solution for a problem of life, in this case of motility or feeding.
Okay,
and I would say that that about the regulation of this, very little is known molecularly, very little. Now there's a project to sequence the genome of Euglena cells, and I guess that in coming years more will be known about it.
Okay, amazing.
So is this size of the pellicle? Sorry, Jen, do you have a question?
Yeah, go ahead. Of course.
I was going to ask if this size of the pellicle, is it consistent between Euglena cells?
Actually, yeah, no. So there's a big variability. Actually, I don't have many pictures here, but here you have a few pictures, right? So this will range between I would say a few hundred of nanometers to maybe a micron, and in this range they will be plastic, so they will be able to slide. So there are Euglenids such as these ones that have lost the ability to have a motile cortical complex. So these are rigid envelopes. So there's a variability, a big variability, and there's a big variability in the shape, even amongst, there's a big variability in the shape that these strips have. And actually this this morphological variability makes of this system a favorite system to study evolution, because I don't know much about it, but I know that when you look at the diversity and the shapes, it's really wonderful. And but the size, for motile cells, it's roughly of a few hundred nanometers.
Yeah, that's cool.
Great. I think we're wrapping up, and we're obviously over time. I just wanted to follow up. There's one more question for Katie that we didn't get to ask, and the participant's still here. So from Yutaka, asking that dynamin-dependent endocytosis is a key to the wound healing. Is there any epistasis between dynamin and shibiri and RAP1? For example, dynamin also accumulates at former TCJs. Is this accumulation quicker or slower than that of RAP1?
Yeah, I think that's a great question, and not something we've yet explored. I don't know any of any links like between RAP1 and dynamin shibiri, but yeah, I would be interested to see, because we're not entirely sure how E-cadherin is getting to the tricellular junctions yet, whether it's through like endocytosis and then exocytosis, or whether it's moving along the membrane, flowing along the membrane to accumulate there. So I think that would help us kind of figure out what's going on. Yeah, it's a great point.
Oh, that's very interesting. And as to the accumulation of E-cadherin, if endocytosis is blocked, it doesn't happen. But I don't know, it's endocytosis, and the accumulation should be exocytosis or diffusion-like thing. And the actual mechanism is not clear, but anyway, it's dependent on endocytosis.
Right, yes, endocytosis definitely initiates initiates the process. So yeah, it'd be really cool to see if there's a link between RAP1 and that process.
Looking forward to your next report. Thank you.
Great, thank you.
We also had a question. Sorry, Ricard, just one second. From Rob Tetley, asking if the RAP1 dominant negative flies, it looked like the actin was preferentially enriched on the dorsal and ventral side of the wound, and asks if the RAP1 dominant negative somehow affects the inherent polarity of the entire embryo.
Yeah, so there's actually a polarization prior to wound healing in this tissue normally. So there are these cables that run along the epithelium. So if you were looking at the movies of the wound, they were kind of these vertical stripes. RAP1 does not seem to play a role in that initial polarization. We still see those cables in both cases, but that could be contributing to the initial heterogeneity that we see. Maybe the initial accumulation is only coming from those cables and there's no additional accumulation. Yeah.
Cool, thanks. Ricard, do you want to go ahead for Marino?
Yeah, thanks. I had another question for Marino, which is, can I think of this surface as an active nematic surface with extensile stresses due to the sliding of the different ridges?
Yeah, maybe yes. I mean, yeah. I mean, yeah. I mean, I don't know to what. Yeah, probably it's anematic with extra constraints, right? So this is an extremely constrained surface. Okay, so basically, I would say that it's if you want to think about it, when you want to describe it in those terms, it's a surface that can shear along directions that are material, that are affected by the deformation, basically, and this would be the definition of it, and how this is mapped into the theory of nematics. I didn't think about it, but there's probably a way to do that. Yeah, it's interesting.
Cool. Yeah.
Okay, fantastic. Thanks everyone who stuck around the extra 15 minutes past the hour. Thank you, Katie, for a fantastic talk. Thank you for Mourino for also a fantastic talk and for pointing out our biases that we have in this seminar series. It's really great to have diversity of speakers.
I'm also very biased in my own research towards animals, so it was just a little provocation. Thank you very much for the invitation on this fantastic series.
Yeah, thank you. No,
and it really, yeah, thank you both. And it really always points out to me just the sort of the wealth of things that I know nothing about in the world, which is exciting and great. All right.